right, sharing my screen. All right, my name's Amber, and the project I'd like to do is studying the gut microbes within monarch caterpillars. Some background, as we all know, Lepidoptera is one of the 5 most diverse insect orders, and the picture here is the monarch butterfly life cycle. I would be focusing on the larval stage, the caterpillar up in the top there. So there's a lot unknown about the gut biome within caterpillars, but the little bit we do know is that it has low volume of resident microbes that the caterpillar itself has. A lot of the microbes that have been studied within caterpillar guts are actually dependent on their diet and the leaves they eat. They take them like directly from that. And this indicates that they probably rely on a transient population, which means the leaves that they eat, the microbes they take in from that, that's what they rely on for their digestion. But other countermeasures the caterpillars have themselves could also be a factor. Caterpillars have a high pH in their gut. It's about a pH of 10, which is very acidic, so bacteria might not survive that as well. They have a very simple structure. It's pretty much in and out for caterpillars. Since they eat and eat so much, they have a fast digestion time because they have to eat and able to eventually get to the adult stage of being butterflies. What I've included here is a chart that I found in one of my primary articles that I was using for background. The red dots are all caterpillars that they took in, and then the others are listed on the key there on what type of animal that they studied. They took the body mass compared to the number of microbes within the stomachs of these animals, and caterpillars all had a relatively clustered number of microbes, while the other species, especially the vertebrates, had very high numbers of microbes within their guts. So there is symbiotic relationships that have been studied between different insects and their gut microbes. So it's interesting that the caterpillars have so few. For example, termites relying on certain gut microbes for a strong source of glucose. American cockroaches have been studied that when certain gut microbes are decreased, it decreases their overall growth and the fertility of the cockroaches. And this is very key because Lepidoptera are pests to some crops, certain species are, and that is why so many studies have been done about them recently and their gut microbes, because they're hoping to be able to develop antibiotics to limit different microbes that would decrease the fertility of these pest Lepidoptera. So that way the pesticides would be specific to that species and it would limit the pest. So a little bit on antibiotics that I've looked at. The fluorocolines and the beta-lactams are 2 types of antibiotics that are known to greatly reduce gut microbes in different species. I've read a lot of studies about them decreasing the ones in humans. I've been talking to Greenfield a bit about different antibiotics I could use. Examples that I'm thinking of fluorocolin and beta-lactams, 2 words I'm definitely going to practice pronouncing. Ciprofloxacin and penicillin are 2 that I'm kind of looking at using now, but I'm definitely going to continue to conference with Gailey to figure out just which ones would be effective and which ones we have access to. The objective of what I'd be doing here is to hopefully further understand the biome with caterpillar stomachs, since not a lot is known, and then through that hopefully understand the symbiosis between caterpillars and their microbes and the few that they do have. And I would hopefully maybe be able to determine the function of different microbes if I'm able to isolate them enough and understand why they benefit the caterpillars. My working hypothesis here is that over half the bacteria I find within the guts of the caterpillars will be directly associated to the plants that they eat. I'm hoping that the antibiotics, though, will be able to help me determine which are from the caterpillar, which are from the leaves, and kind of tie certain ones, hopefully specifically to the caterpillar. My proposed method here, I'll hit some key points here and hopefully hang out long enough where you can pause it to kind of read through it. I'm hoping to have about 15 monarchs per group because that's just a large enough number that I not be just working with two. It'd be a little more statistically sound. I put them in different tanks for the different groups. I thought about housing them as each individual like caterpillar, but that just kind of seems insane, and I would need a lot of containers to put the bugs in, and this way they're all getting the same environmental conditions. I'd hope to have 4 groups, a control group that's not exposed to any antibiotics, one group that's only exposed to ciprofloxacin, and one group only exposed to penicillin, and then one group that's exposed to both to kind of understand how each drug affects them separately together to see if there's any overlap, and then of course the control to just compare everything back to. And to expose them to the antibiotics, I would actually mix them with distilled water to make a very concentrated spray that I'd spray directly on the leaves. And then I'd let them kind of dry and soak in so that when the caterpillar eats the leaf, it's eating actually the antibiotics and it's not kind of running off in water. Then as the caterpillars grow, I would collect their feces. I would monitor their weight to kind of see if the antibiotics are affecting how much their nutrients they're actually taking in to grow. And I'd also like to make notes on their behavior to see if the antibiotics affect any of the typical behaviors. And then when they form chrysalises, I'd want to time how long each caterpillar takes to turn into a butterfly to see if the nutrients intake has any effect on that. And then I would like to note the appearance of the adult butterflies just to kind of see how the antibiotics might be affecting the caterpillars. And of course, at the end of all of this, I'll perform analysis. For anyone wondering what caterpillar poop looks like, I definitely asked this question when Dr. Anderson and Dr. Greenfield brought up the idea of me analyzing the poop. That is a picture of caterpillar poop, and it's definitely big enough that I'll be able to find and collect it. For my data analysis, I'll be using PCR to look at the different microbes. I'll run PCR on the feces. There'll be some poop processing, as I have termed the process, because we're not exactly sure if it'll be refined enough to go through PCR, or if I'll have to. Dr. Greenfield said it might be as simple as just boiling it, or I might have to run some DNA analysis first. But if that doesn't work, if we can't collect enough poop that's viable, I'm probably going to have to sacrifice one of my test caterpillars from each group, maybe 1 or 2, I'm thinking, and actually crush the caterpillar up and run that through the PCR. The significance of this is certain Lepidoptera are pest to crops, so if we can better understand their digestive system, we can make pesticides that are specific to them and won't harm other species, like the study we read at the beginning of the semester, which I guess was only 2 weeks ago. And also there's little currently known, so I think just knowing as much as we can about nature as people will hopefully benefit us somehow, especially with caterpillars since they rely on so few bacteria in their digestion. It'd be definitely interesting to learn more about that, considering us as humans rely on a lot of bacteria to help us break down our food. And then here are my references for the 3 primary sources I used in my background, and the images I showed you are referenced here. Thank you.